Okay, so as I mentioned, today we are starting cardiovascular system physiology, and during this lecture, I'm going to describe you the electrical properties of the heart. As you can see on this graph, there are several types of cells within the heart. The most important from cardiovascular system physiology point of view are cardiomyocytes, the pacemaker cells, uh, and the cardio and the fibroblast. Those are the three most important cell types in the heart. The pacemaker cells, which are present in the basically all part of the conductive system of the heart, including SA node, so sinoatrial node, AV node, atrioventricular node, bundle of His, left and right bundle of His, are characterized by excitability, so they can generate action potential. The pacemaker cells within SA node and AV node can generate action potential spontaneously without a stimuli coming from the central nervous system. Cardiomyocytes also can generate action potential, but only after stimulation coming from the conductive system of the heart. So only once they are stimulated, once the cardiomyocytes are stimulated by the action potential, which is generated by the pacemaker cells. The other feature of cardiomyocytes is contractility. So in response to action potential, cardiomyocytes contract. Fibroblasts, the last type of the cell which we are going to discuss, our response are basic can be determined as can be determined as a tissue which blocks any electrical currents. And because of the fibroblasts present within the heart, the conduction system of the heart must conduct the action potential through the heart. As I mentioned earlier, the pacemaker cells are characterized by the spontaneous electrical activity. In other words, they can generate action potential without a stimuli coming from central nervous system. And this is called heart automaticity. So the ability to generate action potential without a stimuli coming from the central nervous system is called heart automaticity. And how is it possible? What are the mechanisms responsible for the action potential generation without a stimuli? Uh, Contrary to neurons, the pacemaker cells in the heart, especially in the sinoatrial node or atrioventricular node, are characterized by unstable resting membrane potential. Why it is unstable? As any type of cell membrane, the cell membrane of pacemaker cells has many ion channels. In case of pacemaker cells, there are some very characteristic ion channels types and which are responsible for the action potential generation. These channels include HCN channels, T-type, T-type calcium channels, and L-type calcium channels. HCN channels are also known as Thane channels. Thane channels or HCN channels are sodium-potassium voltage-gated channels. Due to electrochemical gradient for sodium and potassium, after activation and opening of the HCN channel protein, we observe the rapid influx of sodium into the cell is observed with reduced outflux of potassium from the cell. Okay, the influx of sodium ions into the cell leads to slow depolarization. Although HCN channels or Thane channels are voltage-gated sodium-potassium channels, they can be also activated by cyclic nucleotides, such as cyclic AMP. The name HCN comes from the factors which activate the channel protein. H stands for hyperpolarization, so negative membrane potential. CN stands for cyclic nucleotides, such as cyclic AMP. Okay, so Thane channels, HCN channels, are activated by negative membrane potential and cyclic nucleotide. The opening of the channel protein leads to sodium influx into the cell with reduced outflux of potassium ions from the cell. This influx leads to cell membrane depolarization, and the depolarization of cell membrane activates T-type calcium channels. The T-type calcium channels open, and therefore we observe an influx of calcium ions into the cell. This influx increases the membrane potential up to the threshold, or around to the membrane potential around threshold. And once the membrane potential is around minus 40 millivolts, L-type calcium channels are activated. The channel protein opens, and we observe the influx of calcium ions into the cell. So, looking at this graph, once the membrane potential is around minus 60 millivolts, which is a resting membrane potential for pacemaker cells, the Thane channels are activated. Therefore, we observe a sodium influx into the cell. This leads to slow depolarization, which activates T-type calcium channels. Due to sodium and calcium influx into the cell, the membrane potential reaches the membrane potential, which is around minus 40, minus 45 millivolts, which is a threshold. And this increase of membrane potential allows to, maybe not allows, but it activates L-type calcium channels. Thanks to the activation of T-type calcium channels and L-type calcium channels, the threshold is reached and the action potential is generated. HCN channels and T-type calcium channels close basically soon after reaching the threshold. So the polarization after the threshold, once the threshold is reached, is due to calcium influx through the L-type calcium channels. So please remember that calcium current before reaching the threshold is due to calcium influx through the T-type calcium channels, whereas the calcium current and calcium influx into the cell after reaching the threshold is due to calcium influx into the cell through L-type calcium channels. After action potential generation, there is a rapid repolarization. The L-type calcium channels close, and then voltage-gated potassium channels open. There are at least two types of potassium channels which are responsible for pacemaker cells membrane potential repolarization: rapid and slow voltage-gated potassium channels. Thanks to potassium outflows from the cell, the membrane potential reaches the value of resting membrane potential, so minus 60 millivolts, and this activates and opens Fanny channels, also known as HCN channels, and the whole cycles goes again and again. So this action potential, which is generated by the pacemaker cells, is the action potential which is responsible for heart contraction. In physiological conditions, the action potential which stimulates the cardiomyocytes to contract is generated by the pacemaker cells within the sinoatrial node. Okay. The action potential which is generated by the pacemaker cells in the sinoatrial node is generated with the frequency around from 60 to 100 action potential per minute, which is actually equal to the physiological heart rate. So sinoatrial node pacemaker cells activity are responsible for the physiological heart rate. The action potential which is generated in the sinoatrial node is then conducted by the intranodal pathways to the atrioventricular node. There are three intranodal pathways, and additionally, the action potential which is generated by the sinoatrial node, which is located in the right atrium, is conducted by the Bachmann bundle over here to the left atrium. Thanks to Bachmann bundle, the action potential, which is generated on the right side of the heart in the right ventricle, is conducted to the left side of the heart and then can be conducted to the all cardiomyocytes in the left atrium. Let's focus on the right side of the heart. As I said, the action potential which is generated in sinoatrial node is conducted by the intranodal pathways, intranodal tracts to the atrioventricular node. The atrioventricular node also has pacemaker cells, which can generate action potential and therefore can regulate the heart rate. The regulation or the action potential generation by pacemaker cells in the AV node is characterized by reduced frequency. The action potential are generated, as you can see over here, 45 to 50 action potentials per minute. In physiological conditions, this electrical activity of the AV node pacemaker cells is inhibited by the overdrive suppression, the mechanism of overdrive suppression, and therefore the action potential which is generated in the SA node, sinoatrial node, is the one which is responsible for the contraction of the cardiomyocytes. The action potential from the AV node is then conducted to the bundle of His, the right bundle of His, left bundle on His, and then Purkinje fibers. As you can see over here on this graph, the bundle of His, the left and the right bundle of His, also have pacemaker cells which can generate action potential. However, the frequency of generated action potential is very low. In case of a bundle of His, it is around 40 to 45 beats per minute. Purkinje fibers can generate action potential with the frequency between 35 and 40 action potential per minute. These frequencies are far too low to maintain proper physiological cardiac output, so the amount of blood pumped by the left or right ventricle during 1 minute. So basically, these frequencies does not allow the heart to pump enough blood to the body. Not only frequency of action potentials are different in different part of conduction system of the heart. The action potential is conducted in different parts of the conduction system with different velocity. As you can see over here, the slowest velocity of conduction is observed in atrioventricular node. In case of SA node, the SA node, the intranodal tract, the velocity is equal around 0.5 meters per second, whereas in AV node, the velocity is reduced to 0.05 meters per second. This reduction of conduction velocity is called decremental conduction, and decremental conduction allows to separate atria and ventricles contraction. The AV node is not only characterized by decremental conduction, but also is characterized by two pathways by which action potential can propagate along the AV node, the fast and the slowed pathway. During normal physiological conditions, during which the SA node, sinoatrial node, is responsible for the action potential generation and the heart rate is equal to the frequency of action potential generated by the SA node, the action potential is conducted by the fast pathway in the AV node. Like almost all action potentials, the action potential of pacemaker cells is characterized by refractory period. Absolute, relative, and in some cases, effective refractory period. Refractory period is, in most of the cell, is a link or is a combination of absolute refractory period and the short period of relative refractory period. So in effective refractory period, some portion of sodium voltage gated channels can be activated. They restore their primary conformation and can be activated, but the action potential cannot be conducted. There are too few available voltage-gated sodium channels. Therefore, action potential cannot be generated. So once the action potential propagates along the fast pathway, the cell membrane of the cell on the fast pathway over here are in the state of effective refractory period. This prevents re-entering the action potential to the AV node by the action potential which propagates along the slow pathway. So action potential is propagated in the AV node by two pathways, the slow and the fast pathway. Because fast pathway allows to conduct action potential with greater velocity, once the action potential propagated by the slow pathway reaches the exit point of the AV node, the action potential cannot re-enter AV node because the cells in the fast pathway the cell membrane of the cells in the fast pathway are in the state of effective refractory period. If there is some kind of a blockage in the AV node, okay, the action potential which is conducted through the slow pathway can re-enter the AV node by the fast pathway, and therefore some kind of a circuit is formed, and this increases the heart rate. And this increase in the heart rate is in most of the cases responsible for the most cases of atrioventricular tachycardia. Is that clear, or do I need to explain one more time?
I think it's clear.
Okay, so once the action potential propagates along the conduction system of the heart, it reaches cardiomyocytes of atrium or of atria or the ventricles. And now this action potential must be, let's call it transferred, must propagate on the cell membrane to the cell membrane of cardiomyocytes, because the cells of the conduction system of the heart lack contractility. Okay, and the propagation of action potential in the heart from the conduction system to the cardiomyocytes or within the conduction of the heart is possible because the elements of the heart, the cells within the heart, are connected by the gap junction. So are connected by the electrical synapses. You had neurophysiology, so I hope that you know what are gap junctions. So this is basically ion channels which connect. Gap junction has an ion channel which connects two cells and allows the ion current to flow from one cell to another. Okay, and the gap junction connects not only cardiomyocytes but also connects cardiomyocytes with the elements of conductive system of the heart. And that's why we may say that the cardiomyocytes function as a one functional syncytium. So once action potential is conducted to a single cardiomyocyte, then this action potential can propagate to all cardiomyocytes within left right ventricle or left right atrium. Once the action potential is conducted to cardiomyocytes, the cardiomyocytes generate their own action potential. Um, so, sorry for the noise. During the action potential of pacemaker cells, we observe a very rapid influx of sodium ions and even greater influx of calcium ions into the cell. Okay, that's why once the action potential is generated in the pacemaker cells, we observe the increase of sodium and calcium concentration inside the cell. The calcium increase is greater compared to sodium concentration increase inside the cell. That's why once the gap junction is activated, the gap junction which connects the cells of the conduction system with cardiomyocytes, we observe a sodium-calcium current to the cardiomyocyte. So, let's say that this is a cell within the conduction system of the heart, which is connected by the gap junction to cardiomyocytes. During the action potential of pacemaker cells, we observe the increase of sodium ions inside the cell and even greater increase of calcium ions inside the cell. That's why in the next step, the sodium-calcium current flows into the cardiomyocytes. The influx of positive charges inside the cell increases the membrane potential. And this increase of membrane potential due to sodium-calcium influx into the cell, mostly calcium, activates fast sodium voltage channels. And this is how the action potential start being generated in cardiomyocytes. So the influx of ions through the gap junction activates fast sodium voltage gated channels, which leads to phase zero of the action potential, so fast depolarization. Due to sodium influx into the cell, the great influx of sodium into the cell, the membrane potential increases rapidly, and once it reaches around plus 50 millivolts, fast sodium channels close, and in the next steps we observe repolarization of the cell membrane. The repolarization of the cardiomyocyte cell membrane is longer compared to the repolarization of neurons or pacemaker cells. It consists of 3 phases: phase 1, phase 2, and phase 3. Once the membrane potential reaches around 50 millivolts, as I said, the voltage gated sodium channels close, and we observe the opening of the potassium voltage gated channels TO1 and TO2. Because of the opening of potassium TO1 and TO2 voltage channels, we observe slow repolarization which leads to phase 2. Phase 2, which is also known as plateau phase. During plateau phase, the membrane potential stays the same. How is it possible? The decrease of membrane potential due to outflux of potassium ions through the voltage-gated potassium channels TO1, TO2 activates L-type calcium channels, and therefore you observe a rapid influx of calcium ions into the cell. At the same time, potassium slow voltage gated channels are activated as well, and therefore the influx of calcium ions into the cell through the T-type calcium channels is balanced by the outflux of potassium ions through the voltage gated slow channels. This phase, so plateau phase or phase 2 of action potential, is a very important phase of the action potential. It not only prolongs the repolarization of the cardiomyocytes, but it also allows calcium influx into the cell. I hope that you remember that cardiomyocytes are characterized by contractility, so they can contract. From the skeletal muscle physiology, you know that the contraction is induced or is due to the calcium influx into the cell, which triggers cross-bridge formation. And it is exactly the same in the heart muscle. The calcium influx into the cell by the L-type calcium channels triggers excitation, contraction, coupling. So phase 2, plateau phase, is the time of action potential when excitation, contraction, coupling occurs. So the amount of the contraction force basically depends on the amount of calcium ions which flow into the cell through the L-type calcium channels. Slowly, the influx of calcium ions through the L-type calcium channels is reduced, and the potassium outflux from the cell causes membrane potential to decrease. Soon after slow potassium channels opening, rapid and ultra rapid potassium channels are activated. The L-type voltage channels close, and this leads to final repolarization of the cell membrane. At the end of the repolarization, K1 potassium channels are activated and opened, and their function similar to the leak channels in the neurons. So basically, they are responsible. The K1 potassium channels are responsible for maintaining the resting membrane potential. And it is obvious that at the beginning of fast depolarization, so over here, we observe a closing of K1 potassium channels by the sodium influx into the cell, and then they reopen again at the end of the repolarization, so phase 3. On this graph, again, you can see the channels and the ion currents during the action potential of cardiomyocytes. Phase 0, fast depolarization due to sodium influx to the fast sodium channels. The opening of fast sodium channels is possible due to depolarization induced by the sodium calcium current or calcium current through the gap junction. Then the depolarization leads to the opening of TO potassium channels, TO1, TO2 potassium channels, which is responsible and which allows potassium outlook from the cell. The membrane potential decreases, phase 1, and then L-type calcium channels. L-type calcium channels open. We observe calcium influx into the cell and outflux of potassium from the cell through, at the beginning, through the TO channels and potassium slow channels. And finally, final repolarization. We observe potassium current through the rapid, ultra rapid, and slow potassium channels. As I said, the cell membrane of the cells, which can generate, excuse me for a moment. Okay, so the cell membrane, which can generate action potential in most of the cells, are characterized by refractory period. In case of pacemaker cells, the whole duration of action potential is associated with absolute refractory period. So during the action potential of pacemaker cells, a second action potential cannot be generated. In case of cardiomyocytes, the situation is a bit more complicated. As you can see on this graph, there are three types of refractory periods in the cardiomyocytes. The absolute refractory period, the effective refractory period, relative refractory period, and the last one, supranormal period, which is a part of relative refractory period. Similar to neuron, during absolute refractory period of the cell membrane of cardiomyocytes, the second action potential cannot be generated. And this, the absolute refractory period starts from resting membrane potential, or we may say starts once the threshold is reached, to, let's say, 1/3 of phase 3. So from here till here, we observe absolute refractory periods, and the second action potential cannot be generated. Effective refractory period is a bit longer than absolute refractory period, so it overlaps a bit with the relative refractory period. During the effective refractory period, the sodium channels become available to carry sodium current. However, there is too few of the ion channels. That's why the action potential cannot be generated. During refractory period, relative refractory period, the channels return to the baseline condition. They have recovered, and that's why the action potential cannot be generated. During supranormal period, which starts at the end of relative refractory period till the and lasts till the cell membrane reaches the resting membrane condition, which is more negative than in case of pacemaker cells, and it is equal to around minus 90 millivolts. During supranormal period, the membrane is more excitable than normal. So basically, during supranormal period, it is easier to generate a second action potential compared to the resting membrane potential. Why? because during supranormal period, all voltage-gated sodium channels recovered, and the cell membrane potential is more positive compared to the resting membrane potential. It is closer to the threshold. That's why it is easier to generate an action potential. As I said, the second phase of the action potential of action potential the action potential of cardiomyocytes, the influx of calcium ions through the L-type calcium channels triggers excitation contraction coupling. As we discussed during skeletal muscle physiology, excitation contraction coupling is responsible for the contraction of the muscle cells due to cross-bridge formation. In case of heart muscle, the excitation contraction coupling starts a bit in a different way compared to the skeletal muscles. Why? The L-type calcium channels, also known as DHP receptors, are not connected by the feet to ryanodine receptors. So in cardiomyocytes, there is no connection between L-type calcium channels, DHP receptors, and ryanodine receptors. No physical connection. Okay, so sarcoplasmatic reticulum. The action potential, which is generated by the pacemaker cells, propagates along the conduction system of the heart, reaches the cardiomyocytes, and this action potential activates DHP receptors, L-type calcium channels. Because of that, we observe calcium influx into the cell, and the calcium influx into the cell through the L-type calcium channels triggers a condition known as calcium-induced calcium release, a condition which is characteristic for the is characteristic to the cardiomyocytes. Calcium ions, which flow into the cell through the L-type calcium channels, bind to ryanodine receptors. Therefore, the channel protein of the ryanodine receptor opens, and we observe a massive influx of calcium ions to the from the sarcoplasmatic reticulum to the cytosol. The calcium ions bind to troponin C. The tropomyosin is removed from the actin, and the cross bridge is formed. Okay. As we said during the skeletal muscle physiology, the influx of calcium ions into the cell starts contraction, whereas the removal of calcium ions from the cell is responsible for the relaxation of the skeletal muscle cell. In skeletal muscle, as well as in cardiomyocytes, there are several structures which are responsible for calcium removal from the cell. The first one is SERCA. So sarcoplasmatic calcium ATPase. SERCA is a calcium pump which removes calcium ions from the cytosol back to sarcoplasmatic reticulum. The activity of SERCA is regulated by the protein known as phospholamban. Unphosphorylated phospholamban inhibits the activity of SERCA, and therefore it reduces the efficiency of calcium removal from cytosol to the sarcoplasmatic reticulum. Once phospholamban is phosphorylated, it dissociates from SERCA, and SERCA can remove calcium ions from the cytosol back to sarcoplasmatic reticulum more effectively. Besides SERCA, calcium ions are removed from the cytosol by calcium pump in the cell membrane. As well, they can be removed by the sodium calcium exchanger. Why I said that they could be removed by the sodium calcium exchanger and not were removed. Sodium calcium exchanger is a molecule which uses electrochemical gradient for sodium ions as a driving force. Okay, so once the membrane potential is negative, like during the resting membrane, like during when the membrane potential is equal to resting membrane potential, the electrodriving force for sodium is directed inwardly into the cell. That's why the sodium calcium exchanger transports sodium ions into the cell and remove calcium ions from the cell. When the action potential is more positive, let's say around plus 50 millivolts, the electrochemical driving force for sodium ions has a different direction. It is directed out of the cell. That's why once the cell membrane is polarized, the sodium calcium exchanger removes calcium ions. It transport calcium ions into the cell and remove sodium ions from the cell. So sodium calcium exchanger can remove calcium ions only when the membrane potential is negative. Okay, once the cell membrane is depolarized, the sodium calcium exchanger transports calcium ions into the cell and removes sodium ions from the cell. The influx of calcium ions to the cell, the transport, maybe not influx, but the transport of calcium ions into the cell by the sodium calcium exchanger helps the L-type calcium channels to deliver a proper amount of calcium ions into the cell. So it supports the L-type calcium channels in calcium transport into the cell. During depolarization, the sodium calcium exchanger supports fast sodium channels. Okay, so as I said, oh, sorry for that, I forgot to mute it. So once the calcium ions flow into the cell by the L-type calcium channels and ryanodine receptors, the calcium ions bind to troponin C. Okay, and because of that, tropomyosin is removed from actin, and now a cross bridge can be formed. To form a cross bridge, the myosin must be in high energetic state. So after ATP hydrolysis. So this is actin after the removal of tropomyosin, and now let's add ATP to myosin. The ATP binds to myosin head, which hydrolyzes ATP which hydrolyzes ATP to ADP and phosphate, and using the energy from ATP to convert to move the myosin head within the neck region. In the next step of cross-bridge formation, the myosin bind to actin, which leads to ADP and phosphate dissociation from myosin. And next, myosin head moves within the region, within the neck region one more time, becoming low energetic one more time. The binding of new ATP molecule causes myosin dissociation from actin, and the whole cycle starts from the beginning. So this is a cross bridge formation. The activity the activity of the cardiomyocytes allows to heart to contract. The contraction of the heart is a oxytonic contraction. Oxytonic contraction is a contraction which consists of two phases: isometric contraction and isotonic contraction. During isometric contraction, the muscle length stays the same. That's why the name isometric, iso constant, metric length. Although during isometric phase, the length of the sarcomere stays the same, the length of the muscle stays the same, the tension is generated. Once the tension of the cardiomyocytes is greater than the afterload, we observe the isotonic phase of the contraction. Okay, and now I think that maybe not now, but during the next lecture, we'll try to figure it out what is the preload and what is the afterload for oxytonic, for the cardiomyocytes, and for the oxytonic contraction of the heart. The activity of pacemaker cells and the cardiomyocytes can be regulated and is regulated by autonomic nervous system. As you can see on this graph, the parasympathetic nervous system innervates mainly the conduction system of the heart, whereas sympathetic nervous system innervates the conduction system of the heart as well as the cardiomyocytes within the atria and ventricles. The parasympathetic nervous system, as you can see on this graph, does not innervate ventricles. So ventricles have no parasympathetic nervous system innervation. Atria are innervated by parasympathetic nervous system. Therefore, the parasympathetic nervous system can regulate the contraction force, directly can regulate the contraction force of the cardiomyocytes within the atria, but it cannot regulate directly the contractility and the contraction force of cardiomyocytes in the ventricles. Okay, so let's focus on the sympathetic nervous system right now. As I said, the sympathetic nervous system innervates both the conduction system of the heart and cardiomyocytes. The cell membrane of the pacemaker cells in the SA node, AV node, and other elements of the conduction system of the heart has many beta-1 receptors. Beta-1 receptors, which you can see over here, are GS-bound receptors. I don't know whether you have ever heard about G-bound receptors. That's why I'm going to discuss the mechanism of its action step by step right now. So, G-bound receptor is a membrane receptor which is connected to the G protein. G protein consists of three types of subunit: alpha, beta, and gamma subunit. During resting condition, the alpha subunit is connected to the G with the G. No, let me think. Sorry, like I have a blackout right now. To the GT or GD molecule. G. Do you know or not? GDP or GTP? Have you ever heard about the G-bound receptor?
I don't think so, Professor.
You haven't. So during the resting condition, it is bound to GTP molecule. Okay. Once the neurotransmitter or neurohormone binds to the receptor, so in case of sympathetic nervous system, it is norepinephrine. Once norepinephrine binds to the receptor, the GTP molecule is replaced by GDP. Okay, so yes or no? Of course, no. The opposite way, sorry. So GDP. Okay, like that. GDP is replaced by GTP, and alpha GTP subunit, the alpha subunit. This, because of that, the alpha subunit dissociates from beta and gamma complex and is released to the cytosol. The beta-1 receptor, which is characteristic for sympathetic nervous system, which is a specific receptor for norepinephrine released by the sympathetic nervous system endings in the conduction system of the heart. In the heart generally, is a GS-bound protein. So this is GS-bound protein. S stands for stimulation. So alpha GTP subunit of GS protein stimulates adenylate cyclase. An enzyme which converts ATP into cyclic AMP. Cyclic AMP is a cyclic nucleotide which can bind to Fano channels. Okay, and because of that, Fano channels open and we observe increased influx of sodium ions into the cell. Furthermore, the increased concentration of cyclic AMP, so one first step, second, third. The increased concentration of cyclic AMP activates protein kinase A. Protein kinase A is an enzyme which phosphorylates different proteins within the cell membrane, like it phosphorylates calcium T and L channels, as well as it phosphorylates slow potassium channels. So in general, norepinephrine binding to the beta-1 receptor within the cell membrane of cardiomyocytes increases the influx of sodium and calcium ions into the cell, mainly sodium and calcium through the T-type calcium channels, and increases the outflux of potassium ions from the cell. That's why the duration of action potential of the pacemaker cells is reduced. More action potentials can be generated per minute, and that's why the heart rate increases after increased activation of sympathetic nervous system. Is that clear? As you can see on this graph, you can see the impact of sympathetic nervous system of the action potential generation in pacemaker cells. Comparing the red line with the blue line, we can see that the depolarization before reaching the threshold over here is much shorter after binding norepinephrine to beta-1 receptor. So the slow depolarization before reaching the threshold is shorter, as well as the repolarization, but mostly the slow depolarization before reaching the threshold is faster. That's why the action potential is generated faster and the heart rate increases. The beta-1 receptor are also present in the cardiomyocytes in the cell membrane of cardiomyocytes. Please remember that the cell membrane of cardiomyocytes is characterized by a very long absolute refractory period. So to increase the heart rate, not only the frequency of action potentials generated in the assay node must be increased, but also the duration of absolute refractory period of cardiomyocyte cell membrane must be shortened. So to increase heart rate, two things must happen. Firstly, the frequency of action potential must be increased, and secondly, the duration of absolute refractory period in cardiomyocytes must be decreased. So by doing these two things, the heart rate can be increased. If we only increase the frequency of action potential generation, the heart rate may not change because of a very long absolute refractory period in the cardiomyocytes. So, as I said, beta-1 receptors are also present in the cell membrane of cardiomyocytes, and they are also GS-bound receptor. So the story goes exactly the same. The norepinephrine binds to beta 1 receptor. The GDP molecule is replaced by GDP subunit. This activates adenylase cyclase, which converts ATP to cyclic AMP and increased concentration of cyclic AMP inside the cell activates protein kinase A. So the activity of protein kinase A increases. And now protein kinase A can phosphorylate and phosphorylates different proteins. The protein kinase A phosphorylates L-type calcium channels, prolonging the opening of the protein channel and increasing the calcium current into the cell. So the protein kinase A phosphorylates L-type calcium channels. It also phosphorylates ryanodine receptors, further increasing calcium influx into the cell. And as you remember from neurophysiology and the skeletal muscle physiology, the greater concentration of calcium ions inside the cell, the greater the contraction force. So the increased influx of calcium into the cell is responsible for the greater contraction force of cardiomyocytes. And this is described as inotropic effect. So sympathetic nervous system exerts inotropic positive effect due to increased calcium influx into the cell. Okay. Furthermore, protein kinase A phosphorylates phospholamban (PLB), which after phosphorylation dissociates from SERCA. It dissociates from SERCA, and the activity of SERCA increases, which results in the faster removal of calcium ions from the cell. The sodium-calcium exchanger can be phosphorylated as well, and during the repolarization phase, the activity of SERCA and sodium-calcium exchanger is responsible for the faster calcium removal from the cell, which leads to the faster relaxation of the cardiomyocytes. So the sympathetic nervous system not only increases the calcium influx into the cell, which leads to increased contraction force and inotropic positive effect, but it also allows to increase the velocity, the speed of calcium removal from the cell. It increases the velocity of relaxation, which is called lusitropic positive effect. So basically, this is all regarding sympathetic nervous system. We are 9 minutes left, and I think that this is not enough to discuss the issue regarding parasympathetic nervous system. So I think that we will finish the lecture a bit earlier. Next, during the next lecture, we are going to discuss the effect of parasympathetic nervous system of pacemaker cells and cardiomyocytes. We'll also try to focus on the effect of sympathetic and parasympathetic nervous system on the conduction system of the heart. So we'll discuss the chronal and dromotropic effect, and then we are going to discuss the cardiac cycle and the blood pressure regulation. So do you have any questions right now? Or no? Okay, so if you don't have any questions, so thank you very much for